Hi, this is Emily, Kendra, and Alexis, and here's how to use a hemocytometer and Countess to count mammalian cells. First things first, you need to subculture in order to get to this point. First, you will have to loosen all the caps. Use a 10 milliliter pipette. Remove 1 milliliter of cell trypsin media solution. And place it into a 1.5 milliliter Eppendorf tube. Take a 1000 microliter pipette or a 100 microliter pipette to take out 100 microliters of trypan blue and place it into the Eppendorf containing your cell trypsin media solution.
Here's the hemocytometer. You're going to go ahead and open it on up. Now inside you're going to have a cover slip that's in a protective sleeve. Take that out, but be careful because it's made out of glass, so it's fragile. It can break very easily. And here's the hemocytometer. It's made out of glass as well. You want to go ahead and dab a Kim wipe with some distilled water, and you want to wipe off both sides of the cover slip. You want to go ahead and use water first, just in case somebody didn't properly clean it. And you lay down a clean Kim wipe, and you're going to do the same thing with the hemocytometer. And then you want to use a new Kim wipe with 70% ethanol. You want to make sure you did the water first before the ethanol, because if you had cells remaining on there and then the ethanol, you will fix them onto the cover slip and onto the hemocytometer. So the water first. Okay, now you need to use the distilled water, and you're going to lightly dab on the outer edges of your cover slip. You don't want to put any water in the center because you don't want to dilute your sample. This is going to help make a seal on the grid of your hemocytometer. You want to go ahead and mix really well your sample that you've added the trypan blue, and you're going to pipette out 10 microliters, and at a 45-degree angle you're going to inject it into the groove of the hemocytometer, and by capillary action it's going to spread underneath the cover slip over the grid marks. You can go ahead and slightly move the outer edge of it if it needs a little help. Okay, you're ready to go ahead and put it on your compound microscope and make sure it's on 10x objective, and go ahead and focus it. The figure on the left shows the counting grid on the chamber of the hemocytometer. There are 9 quadrants total as shown in the figure on the right. we are going to count the 4 corner quadrants. The trypan blue we added is going to stain the non-viable or dead cells blue because their cell membranes are disrupted, which allows the dye to enter the cell. The clear cells are the viable or live cells. Count the clear cells starting from the top of each quadrant from left to right and then down and left like a snake. Include the cells on the top side and right side of the quadrants, omitting the bottom side and left side from your count. This will avoid counting each cell more than once. Use the cell counter to keep track of your count so you don't lose your spot and have to start over. Write down in your lab notebook your cell count for each quadrant.
Now that you have the number of cells in the 4 quadrants, add them together and divide by 4 to get the average. Then multiply the average by 10 to the 4th to determine the cells in 1 milliliter of cell media solution. In order to determine the number of cells in the total 10 milliliter volume in the T75 flask, multiply the cell count in the 1 milliliter calculation by 10.
To clean the hemocytometer, use a Chem wipe diluted in water. First, take off the cover slip and clean the slip. Place on a clean Chem wipe. Use another Chem wipe with water and clean off the excess from the hemocytometer. After water, make sure you use 70% ethanol and clean the hemocytometer slip first. Place inside the casket and then use another Chem wipe to clean off the slide. Place the slide back inside the protective plastic and then inside the case.
Make sure to clean the Countess counting chamber slide like you did before using the hemocytometer. Distilled water first, then 70% ethanol.
Turn on the Countess 2 automated cell counter. The counting chamber slide is very similar to the hemocytometer. To seal the cover slip to the chamber slide, first wet the sides of the slide with water, making sure not to get any in the middle to dilute your sample. Thoroughly mix your sample before adding 10 microliters to the groove of the cell counting chamber slide. Place the counting chamber into the slide holder. Place into the sample port until you hear a soft click. Adjust the focus to see your sample. Click the capture button to view your results. Once you have received your results from the Countess, multiply the live cell concentration by 10 to determine the cell count of the total 10 milliliters in the T75 flask. This step is important when freezing down cells and storing for a long time, because you need to know how many viable cells there are going to be in the cryo vial. For example, if in the future you need to plate 2,000 cells per well plate in a 6-well plate for a transfection experiment, plating non-viable or dead cells would be counterproductive because those cells will neither be able to grow or transfect.